This is the last TRACO lecture for 2015. So for those people who are interested in getting a certificate, the examination will be posted on the website tomorrow, and you can take it any time between now and the middle of January. And you have to get 70% of the questions right to pass. So it's open book, open notes. There's one question derived from each lecture. And I'd like to acknowledge Jonathan Wiest for supporting this course, as well as Anthony Thomas here in Building 50. And up in Frederick, we have Chris Graham. So today, our first speaker is Marina Dobrovolskaia, and she got her PhD in Russia in 1998. And subsequently, she came to the States, did a postdoctoral fellowship at NCI, as well as the University of Maryland. And in 2005, she started working at NCI up at Frederick as a senior scientist, and now she's a principal scientist. The title of her talk, Immunological Properties of Engineered Nanomaterials and Some Challenges in Their Preclinical Characterization.
Good afternoon, everybody, and thank you for joining us this afternoon. Hello, everybody who is watching us online. I'm very happy to be here this year again, share my knowledge and my experience with you. I'm looking forward to your questions. If something is unclear, please feel free to interrupt me. Thank you, Terry, for the invitation and the introduction. So I will be talking about immunological compatibility of engineered nanomaterials and some challenges in their characterization. But before we talk about the immunological properties and characterization of these particles, I would like to go with you over definitions, what is considered as nano. I will be talking about some examples of nanoparticle use in daily life. I will give you some examples of nanoparticle use for cancer diagnosis and therapy, and most importantly, what benefits nanotechnology offers for cancer diagnosis and therapy. We will talk about nanomedicines. I have to say that when it comes to definitions, some people call these materials non-biological complex drugs, or NBCD. This terminology primarily is used in Europe. In the US, we tend to call it nanomedicines, but as you see, the field is evolving. So you may see in future, in reading articles, you may see one definition or the other, but you will know that they refer to the materials that are intended for use in biological applications. I will tell you about NCL, which stands for the Nanotechnology Characterization Lab. Very briefly tell you who we are and what we are doing. And then the majority of my presentation will be devoted to reviewing the information about the biological compatibility and specifically particles' effects on the immune system and their characterization. What is nano? There is no universal definition. Most commonly, people use National Nanotechnology Initiative definition, which speaks of nanoparticles as of objects with a size in at least one dimension between 1 and 100 nanometers. However, if you look at the nano-sized materials that are approved for clinical use and are used in patients, then most commonly these materials exceed 100 nanometers. And for this reason, United States Food and Drug Administration defines any material that exhibits properties or phenomenon, including physical and chemical properties or biological effects that are attributable to its dimensions, even if these dimensions exceed 100 nanometers. So FDA considered as nano up to 1 micrometer. There are a lot of nanoparticles in daily life. According to the web, there are more than 800 manufacturer-identified products from over 400 companies in over 20 countries worldwide. Nanoparticles are used in clothing, wound dressing, washing machine liners, as lens coatings in sunglasses, sporting equipment. Nearly all translucent sunscreens contain nanoscale titanium and zinc oxide particles. Those especially women who like cosmetics should know and perhaps know that these products contain liposomes and emulsions. And some materials, especially carbon-based materials such as carbon nanotubes, are used as structural materials due to their durability. What can nanotechnology offer to those of us who work in drug delivery? Nanoparticles may improve solubility of hydrophobic drugs, and therefore, in drug delivery, one of the most common use of nanotechnology carriers is to deliver hydrophobic drugs. Nanotechnology offers multifunctional capabilities. Here you see the example of nanoparticle, which has a core particle, has a drug bound to the particle surface, polyethylene glycol coating, which makes particle surface stealth or prevent it from recognition by the immune system. It may carry imaging agent, it may carry a targeting molecule. Nanotechnologies that carry both drug and imaging agents are often called theranostics, from the combination of two words, therapeutic and diagnostics. Nanotechnology can also offer tumor targeting and reduce toxicity, and also robotic tasks such as sensing, computation, actuation, and triggered responses. For cancer therapy, nanoparticle offer two different type of advantages. One is called EPR effect. EPR stands for enhanced permeability and retention. What does it mean? It refers to the leaky vasculature of the tumor blood vessels. So the blood vessels around tumor have fenestrations, and nanoparticles, due to their nano size, may cross this leaky vasculature and enter into the interstitial space. And due to the low lymphatic drainage, these materials tend to stay in the tumor space and then release drugs bound to the particle surface. This type of targeting is called passive targeting, because basically particle is utilizing natural property of the tumor and the vasculature to enter the tumor space. There is also so-called active targeting. This is then nanoparticle surface is modified with a targeting agent. It could be monoclonal antibody, which recognizes tumor-specific antigen, or it could be aptamer or any other molecule which specifically binds to tumor-specific antigen. It is important to recognize that in order for the active targeting to work, passive targeting is needed, because before particle can utilize its targeting ligand, it still needs to cross the vasculature and enter into the interstitial space. As I mentioned to you in the beginning of the lecture, in Europe, a lot of medicinal nanoparticles are referred to as NBCD, or non-biological complex drugs. In the US, most common definition that you may hear is nanomedicine. So I will be calling these materials nanomedicines for the purpose of my presentation. What you see on this slide is a classification of the nanomedicines by category. And in categories, we look at the medical devices and we look at drugs. There are two categories. One in investigational stage. So these are the materials that didn't reach a commercial stage yet. They are in various phases of preclinical and clinical development. As you see, majority, according to the literature, majority of these materials are in medical devices category, and only 17% is in drug category. Completely different picture is seen when we look at the materials that already reached commercial stage. 67% of these are represented by drugs formulated using nanotechnology, and 33% are represented by medical devices. What types of nanomaterials are used for drug delivery and in medical devices? Again, what you see from this slide is the data from the literature, and there is a difference between investigational and commercial materials. In investigational materials, you see that the majority of these materials are liposomes, followed by polymeric nanoparticles, emulsions, and solid nanoparticles. In commercial stage, the dominant particle is solid nanoparticles, followed by composite materials. Most nanomedicines are developed for cancer therapy. So the primary market is cancer therapy, and you see it on this slide. Majority of nanomedicines are intended for intravenous administration and have size from 100 and 350 nanometers. So they're roughly less than 350 nanometers in size. Most of these materials are neutral, have hydrophilic surfaces, and spherical. The neutral nature and the hydrophilic surface is important to prevent particle from off-target toxicities and from immune recognition. And I will be talking about this and show you some case studies later in my presentation. There is increasing funding. There are a lot of efforts from funding agencies, the DoD and HHS, and you see the increase in funding nanotechnology research over the years, starting in about 2001 up to 2015. Right now, in the US, we spent about $1,500 million for research in nanotechnology. But the primary purpose of this research is to develop nanoparticles for medical applications, for cancer specifically. But in drug development, there is this definition, the so-called value of death. What it is? Value of death refers to the stage in material development from the moment it was discovered and showed that it has potential to cure cancer or benefit the cancer therapy, to the stage where this material is actually ready to enter into clinical trials. Because there is a lot of the infrastructure, a lot of the knowledge and which is involved in translating these materials from bench into the bedside that requires collaboration between academia and industry. Our laboratory, Nanotechnology Characterization Lab, was established in 2004 as interagency collaboration between National Cancer Institute, National Institute of Standards and Technologies, and US FDA. And the mission of the lab was to accelerate the translation of the promising nanotechnology concepts from bench to bedside. What you see on this slide is a concept of operation. NCL receives material from government organizations, big pharma, academic PIs, biotech companies in the US and also from other entities outside of the US. We subject these materials to rigorous characterization, looking at the physicochemical parameters and also at in vitro pharmacology, toxicology, and immunology. And the final goal of this characterization is to provide materials to our clients to support their regulatory finding, requesting clinical studies of these materials, and also to inform regulatory agencies such as FDA about trends in nanoparticle biocompatibility, about the applicability of traditional methods to nanotechnology formulated drugs. Nanotechnology expertise and resources in multiple disciplines are brought together in one location to serve all of the nanotech oncology researchers. Recently, we also started supporting non-oncology applications. We have been in operations for over 10 years, and during this time, we characterized about 300, a little bit over 300 different nanoparticles. With majority of them were liposomes, polymers, metal oxide, metals, but we also characterized other materials, including dendrimers, emulsions, nanorods, nanowires, quantum dots, carbon nanotubes, nanocrystals, and other types of materials. It is now very well established, both from research in our laboratory and also in the literature from publications by other laboratories, that it is nanoparticle physicochemical properties such as particle charge, size, hydrophobicity, and presence of the targeting moieties that determine particle interaction with biological systems and specifically interaction with immune system. On this slide, I will show you some of the examples. You see in this case is increase in size in PAMAM dendrimers results in increase in the particle reactivity with membranes of platelets that causes platelet activation and aggregation. So in this example, I show you that size is important. The larger the particle, the greater the platelet aggregation. But it is important to recognize that particle size under pristine conditions and in biological environment are different things. Please look at this data. We are using here 30 nanometer colloidal gold nanoparticles in PBS. There is no biological matrix in this system. And we use 3 different methods to look at the particle size: transmission electron microscopy, atomic force microscopy, and dynamic light scattering. And what you see on these yellow rectangles is the measured size. So the 30 nanometers is nominal size, theoretical size, and this is what instruments measure. So about 30 nanometers, right? There is agreement between different methods. Look what happens if we study these particles after incubation in human plasma. Transmission electron microscopy and atomic force microscopy, which look at the core, at the solid core of the particle, still show us the 30 nanometer size. But then we use dynamic light scattering, which shows hydrodynamic size of the particle almost doubles. We now see particle size increases to 76 nanometers. Why does it happen? In plasma, there are a lot of proteins. And these proteins can bind to the particle surface, and the changes, particle gets larger due to the protein coating. And this soft size is not detected by TAM and AFM, but is detected by DLS. And this is the size at which these particles, when they are used in biological applications, are seen by our body. So our body does not see 30 nanometers. Our body will see 76 nanometer particle. Particle surface charge, as I mentioned, is also important determinant of particle biocompatibility. Again, I will use as example platelet aggregation assay, and my model particle in this case is PAMAM dendrimer. This study was done in collaboration with Dr. Anil Patri. What Anil did, he used the generation 5 PAMAM dendrimer, which we knew activates platelets. It activates platelets and induces platelet aggregation. It's a very potent activator. And he used the chemical modification to mask 25% of the particle surface amines, then another 25%, and another 25%, and so forth, till no surface amine left. And as you see, as we decrease the percentage of the surface amine, the reactivity of this particle with platelet decreases. So if we want to use this nanoparticle, if we want specifically to induce platelet aggregation, then we want to use this particle, right? Because we know that it activates the platelets and biologically reactive. But if we intend to use this particle in biological applications and inject it in animals, this material would be toxic. But by masking surface amine, we make it more biocompatible and less toxic. Composition of nanoparticles is also important. This is example where we use triazine dendrimers and PAMAM dendrimers. What are the dendrimers? Dendrimers are also sometimes called artificial proteins. These are globular polymeric materials that if you think of a tree, so this basically globular tree crown. This is spherical material, very soft, which may be made of different building blocks. PAMAM dendrimers are made of polyamidamine, and the triazine made of triazine blocks. We see the same trend in both particles. The particle size increases, the density of surface group increases, we see greater biological reactivity. But we see that the triazine dendrimers are less reactive than PAMAM dendrimers. And the way how we can explain it, at the same generation, the density of the surface group in triazine dendrimers is lower than in PAMAM dendrimers. Also, these materials have different hydrophobicity, which also contributes to their different biological reactivities. Nanoparticles' immunotoxicity may be good or bad, depending on whether it is desirable or not. Some nanoparticles, engineered nanoparticles, are created to specifically interact with the immune system to benefit vaccine efficacy, for use in cancer immunotherapy, to be used in therapy of inflammatory disorders, autoimmune disease therapy, and infection disease therapy. However, there are some nanomaterials that are mostly environmental materials, which surface is not passivated, but they are interactive with the biological membranes and immune cells. And these materials cause undesirable immunotoxicity, leading to hypersensitivity reactions, anaphylaxis, coagulopathy. They may lower body's defense to pathogen and cancer, and also suppress bone marrow and thymus function. What we at NCL are interested in, we are interested in the engineered nanomaterials that are developed for biological, the medical applications, but not specifically to target the immune system. So what we want to know, these materials that are intended for, let's say, cancer therapy to deliver cytotoxic drugs, would they also have some undesirable side effect, which may lead to anaphylaxis or other consequences, undesirable consequences for patient? What we can say from our experience is that nanoparticles indeed can be immunotoxic. However, there is no new toxicity which would be attributed to their nano size. Nanotechnology-based pharmaceuticals intrinsically are not more immunotoxic than other drugs in current clinical use. Moreover, there is increasing number of examples showing that incorporation of the traditional drugs onto nanotechnology platforms helps decreasing their immunotoxicities. And I would like to share two examples with you. One from small molecular drug and one therapeutic protein. So what you will see on this side of the slide, I will show you the traditional formulation of this drug. And then on this side of the slide, I will show you the nanotechnology formulated drug. Doxorubicin, as many of you know, is very commonly used for cancer therapy. This drug can activate endothelial cells and cancer cells to express procoagulant activity complex, which serves as a platform for assembly of the plasma coagulation cascade, which induces plasma coagulation and as a result blood clotting, and this side effect is called disseminated intravascular coagulation or consumptive coagulopathy. Specifically, this side effect was described in acute melanocytic leukemia, so in blood, in some types of the blood cancers, and in these cases, the therapy of doxorubicin had to be discontinued. Then the same drug was encapsulated into pegylated liposome. This formulation was called Doxil. No such toxicities were described. And we have in vitro study showing that induction of procoagulant activity by doxorubicin is confirmed, but there is no such toxicity with pegylated liposomal doxorubicin. Another example is therapeutic protein, tumor necrosis factor alpha. TNF alpha is cytokine, so it is expected for it to be immunostimulatory, but the name of this protein states that it may cause necrosis of the tumors. So no surprise, this protein was studied in clinical trials for cancer therapy, but investigation of this drug was halted by systemic immunological stimulation and toxicity. So in order to reach the dose of this drug enough to kill the tumor, the doctors have to administer so high doses of this material that the patients could not tolerate. So this protein was discontinued. Small biotechnology company in Rockville, Maryland, used the same protein and attached it to the surface of colloidal gold nanoparticles. And they also added polyethylene glycol to mask the immune recognition of the particle surface and the protein that this particle carries. And this product is called Cyte 6091 or Orimmune. This material successfully passed phase 1 clinical trials without reaching maximum tolerated dose. Obviously, then these nanotechnology formulated drugs are entering, before these nanotechnology formulated drugs are entering clinical trials, they have to be rigorously characterized. And this preclinical characterization involves multiple areas of research. It includes chemistry, which analyzes particle size, charge, shape, composition, surface coating, ligand density, solubility, stability, architecture, excipients, anything that is relevant to the chemical composition of the particle and the chemical structure of the particle. There are a lot of challenges in studying efficacy of the nanotechnology formulated drugs because in nano formulations, we have to look not only at the end result, but we also have to correlate it with drug loading, drug stability, drug release, and the targeting. There are a lot of challenges in pharmacology, toxicology, which includes change in particle biodistribution, and I will show you example on my next slide, use of appropriate models, investigation of the toxicity and the mechanism of toxicity. And there are a lot of challenges in hematology and immunology. Some of the common challenges include biological contaminants or biological impurities, which I will talk today in my presentation. Correlation between in vitro and in vivo methods is another subject which I will cover in my presentation. And the predictive methods and development of predictive models to understand nanoparticle immunotoxicities. This is the example of the challenge that many researchers face when they take the traditional drug and they use nanoparticle for delivery of this drug. Again, I'm using doxorubicin because it's very well-studied drug. Many people are familiar with it. It's a small molecule, and typically it distributes to bone marrow and heart. And as you know, the dose-limiting toxicities of the doxorubicin hydrochloride are myelosuppression and cardiac toxicity. Then this drug was reformulated using pegylated liposomes. The distribution to bone marrow and heart decreased, so these two types of toxicities were resolved. However, liposomal doxorubicin tends to accumulate in skin, and side effect is PPE, or palmar-plantar erythrodysesthesia. Both of these materials are used in clinic for cancer therapy. This is experimental preclinical formulation of doxorubicin using cyanoacrylate nanoparticles. Cyanoacrylate nanoparticle formulated doxorubicin did not distribute to skin, so the researchers were very happy they overcome PPE. It did not distribute to bone marrow at the doses enough to cause myelosuppression and heart tissue, so that myelosuppression and cardiac toxicity were resolved. But due to their small size, these particles were accumulating and delivering drug to kidney. So as such, the toxicity relocated from skin to kidney. And this is a very common challenge for many of the researchers who work with formulating of the drug with the purpose of solving one toxicity, because they basically have to monitor the particle biodistribution and see if toxicity relocates from one organ to another due to the change in particle biodistribution and in the drug biodistribution. I also mentioned to you that one of the challenges is associated with the presence of excipients in formulation. So when we talk about nanoparticle nanotechnology formulated drugs, we are not only talking about drug toxicity and the nanoparticle toxicity, we also have to consider other materials that are present in formulation, excipients, which could be detergents, which could be to prevent particle from aggregation, which could be proteins or some cryoprotective reagents to prevent particles from aggregation and improve their stability during lyophilization and storage. In this case, we are using drug-loaded nanocapsules, and then this material was studied in the biological assays and hematology assays. We observed that this material was hemolytic. Why these nanoparticles are hemolytic? The physicochemical characterization, again, we are using dynamic light scattering, did not show any small peaks. It shows that average particle size is 165 nanometers, which was expected, but transmission electron microscopy with negative staining shows presence of the small micelles. We found out that in order to prepare this formulation, detergent Bridge 78 is used. Then Bridge 78 is used to formulate the drug. It becomes non-reactive, but leftover of this material, unreacted Bridge 78, forms micelles. And then concentration of the Bridge 78 is above CMC. We see these micelles, same as we see in the formulation, but if it gets diluted, below the critical micellar concentration, these micelles disappear. It is very well known, as you know, that detergents at the concentrations above CMC are reactive with biological membranes and lyse biological membranes. So that unreacted breach is causing hemolysis in this case. Nanoparticle size and zeta potential are important determinants of particle toxicity, and I showed you some examples of that. But I would like to say that despite of their importance, they are insufficient to discriminate between nanomaterials displaying different biological effects. And to prove this point, I would like to use this case study of the pegylated liposomal doxorubicin. This formulation is known as Doxil. Worldwide, there are different formulations. There is Doxil in the United States. There is Lipodox, which was imported into the United States from India under executive order for several months to provide this important drug to the patient. And now we have also generic version of the pegylated liposomal doxorubicin. In Europe, Doxil is called Calyx, and cyanodoxazome is another version of the pegylated liposomal doxorubicin available in Iran, and this is Lipodox available in India. I will be talking about these three formulations because they were available in the US and we studied these materials. You see the size and the zeta potential of what we call old Doxil, new Doxil, and Lipodox. Just to give you some background, Doxil was available in the US until 2008 when the facility manufacturing this product had some problems with compliance with GMP requirements, and that in compliance, some issues resulted in shortage. So they were temporarily unavailable to manufacture these materials, and many patients were undergoing therapy with Doxil. Doxil gave better results and was preferred by both physicians and patients, but there was not enough Doxil on the market. So under these conditions, United States President issued the executive order to bring Lipodox into the US to temporarily cover this shortage. And meanwhile, the manufacturing procedure and facility were optimized to renew the production of this material. So then this material was produced in the US. It's produced also as Doxil, but we call it new Doxil because new Doxil means after shortage. Okay, and Lipodox is generic Doxil and the Doxil which is imported into the US from India. The important message from this slide is that there is no difference between these formulations in size by DLS and zeta potential. But let's look at its biological reactivity. According to the literature about clinical use of Doxil and also animal studies in pigs model of CARPA, many investigators reported that the old Doxil is inducing complement activation and complement activation related to the allergy. And you see this in vitro assay data are in agreement with those observations. However, new Doxil and Lipodox do not have this toxicity, which again correlates with the clinical observations. Physicians that used Doxil after the shortage noticed that lower incidence of CARPA, and there is a lower propensity of this material to induce complement activation. Why does this happen? So this is a positive change, right? We had a greater incidence of complement activation related to the allergy with the when the procedure was changed, the new material was manufactured, there is less CARPA induction and less complement activation. What we can say that this is not due to any change in particle size and particle zeta potential. There is something else. And we are currently investigating the difference to understand the mechanism. Another challenge related to particle characterization in preclinical stage is contamination with endotoxin. Endotoxin is component of gram-negative bacterial cell wall. It is very common contaminants in nanomaterials, In fact, around 30% of preclinical formulations are contaminated with endotoxin. Endotoxin can be present in nanoparticle suspensions or solutions, or it can bind to particle surface. Especially cationic materials are prone for endotoxin contamination because endotoxin is anionic. So just due to the electrostatic interaction, anionic endotoxin binds to the surface of the cationic particles. Why it is bad? Endotoxin may result in erroneous data and lead us to wrong conclusions. It may confound results of efficacy and toxicity studies. Now there is also increasing data showing that some nanomaterials are not immunostimulatory themselves, but they exaggerate endotoxin-mediated inflammation, and it may cause undesirable potential problems with undesirable immunogenicity of protein-based APIs or targeting ligands. What I show you on this slide is an example of this exaggeration. This is the induction of leukocyte procoagulant activity by pure LPS, and LPS is an important component, is main component of the endotoxin. And alone, the red line, and the blue line shows the effects of the same concentration of LPS in the presence of one concentration of nanoparticle. This concentration of nanoparticle is unreactive. It doesn't cause procoagulant activity induction. So you see that we are looking at the enhancement. The grand challenge in nanomedicine is detection of endotoxin, accurate detection of endotoxin, because many nanoparticles interfere with one or more LAL formats. What you see on this slide are two main types of interference. One is called inhibition, which is defined as spike recovery less than 50%, and another type is called enhancement, which is determined by spike recovery over 200%. What does it mean? Look at this data. This is endotoxin spiked in water. Blue line shows the theoretical value. Theoretical means that we spiked 0.4 EU per milliliter of water, and we recovered almost 100%, right? This is accurate detection. Same in this case. But in this case, we are using colloidal gold nanoparticles. We spike same amount of endotoxin, and now we detect about 0.1 EU instead of 0.4 EU. So this is inhibition. It's less than 50%. In this case, it was polymeric nanoparticle. Instead of 0.4 EU, we now detect 1.8 endotoxin units per mil. So looks like this material contains a lot of endotoxin, but in fact, this polymer behaves like endotoxin in the LAL assay. And this inhibition is very common for cationic nanoparticle, unfunctionalized carbon nanotubes, protein-rich formulations, anionic metal colloids, surfactant-containing formulations. And enhancement is very common for polymeric nanoparticles, and depending on the concentration of protein and surfactant, it can also be observed in protein-rich formulations and surfactant-containing formulations. The reason I would like to share with you next two slides is to show you how tricky it gets sometimes to distinguish between the assay interference and the presence of endotoxin in formulation. What we do in our laboratory, we developed a decision tree according to which we use, for the same type of nanoparticle, we use two different LAL formats. In this case, we use a chromogenic LAL and turbidity LAL. And you see that one assay tells us that this formulation contains 0.5 endotoxin units per milliliter, and this assay shows us that we have 21 endotoxin EU per milliliter. How do we know which one is correct? Inhibition enhancement control, which I showed you in the previous slide, was acceptable in both assays. We use a biological test to verify LAL findings, and this biological test is conducted in rabbits, and it's called a rabbit pyrogen test. Basically, what this test means, you inject material into rabbits and you measure their temperature. And if they develop fever, then it indicates that material contains endotoxin. The material was pyrogenic, so obviously we confirmed the turbidity data. We sent this material back to investigator from whom we received this material. They conducted purification. We retested purified material by LAL, and you see that levels of endotoxin substantially decreased. We repeated rabbit pyrogen test, and we found that material is not pyrogenic. Sometimes it is desirable to conduct biological test, but the test cannot be conducted in rabbits. In this case, we use in vitro assay, which is called monocyte activation test. This test is conducted in monocytes, and we are looking at the induction of the pyrogenic cytokines, TNF, or interleukin-1 and interleukin-6. Here you also see different commercial formulations of nanoparticles, say Abilset, Abraxane, Visudyne, and Ferrahim. And you see that for some of them, like for Abraxane, there is consistent data between different LAL formats, turbidity, chromogenic, and gel clot. But for some formulation, like for Visudyne, the result is inconsistent. And depending on which assay you use, you may either overdose endotoxin or be safe. This data can be verified by monocyte activation test, but there is a nuance, and this nuance deals with the drug that these particles deliver. While Abilset and Ferrahim could be reliably retested in monocyte activation test, and you see spike recovery is acceptable, Abraxane and Visudyne cannot be tested by this test because they contain cytotoxic drug which kills the cells, and therefore the results are false negative. Nanoparticle sterilization represents another challenge in development of these materials because sterilization methods that are traditionally used can change integrity of the particles. In this case study, I review effects of gamma irradiation on citrate-stabilized gold colloids and citrate-stabilized silver colloids. Before gamma irradiation, you see that both particles have a spherical shape and well dispersed. After gamma radiation, the dispersion and the shape of gold colloids does not change, but silver colloids disappear. Instead, we have this wire-like materials formed. Gold colloids are sterilized by gamma radiation. In fact, National Institutes of Standard and Technologies produce standard reference materials that are sterilized by gamma radiation. In case of the silver colloids, not only the shape of this particle changes, integrity of this particle is affected, but their biocompatibility is also affected. You see, the particles before sterilization do not react with platelets and do not cause platelet aggregation, but after sterilization, and as we increase the dose of gamma radiation, these materials change and then they become reactive with platelets. So we have to be very careful in selecting sterilization method which would not affect integrity of the particles and which would not harm drugs that the particle carry, and also which would not change biological properties of these materials. The likelihood of identifying immunotoxicity increases with progression from preclinical to the clinical phase, and the correlation between in vitro and in vivo method is a very hot topic because high cost and ethical issues and low throughput of in vivo tests limit application of these methods. On the other hand, in vitro tests have high throughput and lower cost, but the question which many researchers struggle is whether or not preclinical in vitro tests are predictive. Can we rely on preclinical in vitro tests? I would like to step back a little bit from nanotechnology area and look at the biotechnology. What lessons we learn from biotechnology-derived therapeutics. And in this example, I will review this product called TGN-1412. It was manufactured by the European company TGNIR. What this product is, it's a super monoclonal antibodies that activate CD28. You know, the antigen-presenting cell presents antigenic peptide to T cells through the TCR receptor. T cell receives signal 1, means that it received the signal, there is an antigen, I have to be ready to proliferate, but T cell will not start proliferating until it receives signal 2. And this signal 2 is delivered by interaction between CD28 on the surface of the T cell and co-stimulatory molecules on antigen-presenting cell, CD80, CD86. So these monoclonal antibodies were developed to overcome energy of the T cells, to basically stimulate T cells under immunosuppressive conditions. And then these antibodies were studied in preclinical studies in non-human primates and in rodents. Everything was fine. There was no cytokine storm. However, it was a tragedy in Europe. Then 6 out of 6 volunteers, so human patients who received this drug, developed multiple organ failure and experienced cytokine storm. What you see on this picture is fingers, and many of these patients had necrosis in their toes as well, due to the overproduction of the tumor necrosis factor and other inflammatory cytokines. Interestingly, then these studies were stopped and material was investigated. It was found that this material passes all pyrogenic tests in terms of LAL. It was not endotoxin, it was no chemical or biological contaminants in this material, but the material itself induced cytokine storm in vitro. Then normal human PBMCs, peripheral blood mononuclear cells, were used as a model. Apparently, non-human primate and rodent models were insensitive to this change. So what it tells us that in some cases, you always need to use animal models, because especially with nanotechnology, animal models allow to look at the biodistribution of the material. In vitro models do not test for biodistribution. But in vitro models, having sensitive in vitro models and predictive in vitro models is very important, because it may help to overcome these accidents, these tragic accidents. When it comes to nanotechnology, we now know from our research and also from other studies that common markers for acute nanoparticle toxicities are hemolysis or damage to erythrocytes, complement activation, thrombogenicity, either through activation of platelets or induction of procoagulant activity or both, pyrogenicity, and cytokine induction. Most of these toxicities can be rapidly assessed in vitro prior to more resource and time-consuming in vivo studies. And what I summarized for you on this slide are assays, in vitro assays, and then in vivo assays with good predictability. Good meaning that if you see positive response in vitro, likelihood of experiencing this toxicity in vivo is very high, and fair toxicity. Why I'm saying fair is because usually based on one in vitro study, it's very difficult to conclude, because remember, in vitro models do not allow to account for particle biodistribution. For example, CFU-GM allows us to look at the bone marrow toxicity, which would correlate with in vivo with myelosuppression. So we may see decrease in CFU-GM counts in vitro, because in vitro there is only bone marrow cells and the particle. But if we inject this material in vivo, it may not distribute to the bone marrow. So then in vitro results become irrelevant. But if material does distribute to the bone marrow after in vivo, after IV administration in vivo, then in vitro data correlate with in vivo data. This is a case study showing how nanoparticle-induced cytokines correlate between animal models and in vitro assays using PBMC. In this case, we study metal oxide nanoparticles which have identical metal core, identical core composition, but different surface coatings. The hydrodynamic size and the zeta potential of these two formulations, which I call nanoparticle formulation 1 and nanoparticle formulation 2, was very similar. But then these materials were studied in rodents, in rats, and in rabbits. Then one formulation was very toxic, and necropsy of the animals revealed congestion and multiple organ damage similar to that seen in septic shock. However, when both formulations were tested for endotoxin, they were found to be endotoxin-free. Analysis of the plasma samples from the animals that displayed congestion and spleen showed high cytokine levels. And this is in vitro assay results using human peripheral blood mononuclear cells. Formulation which was toxic in animals induced TNF, IL-1 and IL-8 in human PBMC, and formulation which was not toxic in animals was negative in this assay. So again, this is a confirmation that perhaps among other in vitro tests, peripheral blood mononuclear cells are very sensitive predictors of the cytokine storm reactions. And this is why I would like to spend the rest of my presentation to my other examples speaking about cytokines. What we found very interesting observation. We made very interesting observation by studying cytokine response to nanomaterials. What we observed is among particles that induced cytokines, over 60% of these materials induced interleukin-8. Those of you who do immunology, do we have anyone who does immunology research in this audience? Raise your hand if you do. So then I'll ask you, normally when you speak, for example, about bacterial contamination or bacterial ligands, so like lipopolysaccharide, then you add it to your cells in vitro culture, so in animals, you see multiple cytokines, right? You see interleukin-1, you see TNF, you see interleukin-6, interleukin-10. You don't see, in response to traditional inflammatory stimulus, you don't see induction of interleukin-8 without induction of TNF and IL-1. With nanoparticles, what was striking for me is that we came across nanomaterials that induce interleukin-8 without inducing TNF and IL-1. So about 50%, like roughly 53% of these materials were exclusive inducers of the IL-8. Exclusivity means they induced interleukin-8 without induction of TNF, without induction of the IL-1. And the common denominator between these materials was their composition. Materials that induced interleukin-8 without induction of TNF and IL-1 were liposomes and emulsion. And Anna Ilinskaya in my group did the mechanistic study to show that this induction of interleukin-8 is related to oxidative stress, which did not involve gene expression, but involved protein expression from the pre-existing mRNA. Basically, oxidative stress led to stabilization of the low levels of mRNA that mononuclear cells maintain, and resulted in expression of the protein from this mRNA pool, which was very interesting. Why it is relevant for us to look at the cytokine storm? Because I think by now you are convinced that it is easily to predict in vitro, and it is a common toxicity. Some therapeutic products, for example, therapeutic oligonucleotides, are known to induce cytokine storm, and specifically they induce interferons, type 1 interferons and type 2 interferons. Then this, now we have nanocarriers that induce inflammatory cytokines. If we use these nanocarriers that induce cytokines for delivery of therapeutic oligonucleotides that induce interferons, what do we have? Cytokines plus interferons will be induced by the final product. And this is an example. You see, we tested liposomal formulation of antisense oligonucleotide, and nanoparticle platform did not induce interferon gamma. The drug, the oligonucleotide, did. Obviously, we saw interferon gamma induction by the final product. When it comes to interleukin-8, the API, the nucleotide, did not induce interleukin-8, but the platform did, and again, the final product induced interleukin-8. So the bottom line, that final product induces both interferons and cytokines. That led us to create this take-home message for the drug delivery using nanoparticles. This message is very simple, that immunotoxicity of both drug and the nanocarrier have to be considered. And use immunologically reactive carriers, then immunomodulation is wanted, and avoid such carriers, and immunoreactivity is undesirable. I just walk you through this slide. Basically, we are considering immunological properties of a nanocarrier and immunological properties of API to identify common or overlapping toxicities. For example, both carrier and the drug induce cytokines. And if we intend to use this formulation for systemic administration, in which we don't want any immunostimulation, then we have to stop and reformulate this drug using different platform. But if we are talking about vaccine, for example, delivery, or some intended immunostimulation, then overlapping cytokine induction by platform in a drug may be desirable, right? So we can proceed. And also, there is plenty of data in the literature that some nanoparticles may, by composition and through their physicochemical property, may, for example, inhibit toll-like receptor signaling. They may inhibit or prevent complement activation, can inhibit or prevent thrombogenicity, cytokine effect. So these properties of such particles can be leveraged for systemic administration of drugs that are not intended for overt immunostimulation, because they will help to reduce immunotoxicity. In summary, I would like to emphasize several points, one of which is that each nanoparticle, each nanotechnology platform is unique. And therefore, comprehensive physicochemical characterization is very important, not just DLS and zeta potential. The key factors in preclinical development that are important to be understood and thoroughly characterized include physicochemical properties such as particle size, charge, composition, functional properties such as stability, drug loading, drug release targeting, structure-activity relationship. Why do we need to understand structure-activity relationship? If, for example, we develop nanoformulation and we show that it passes all of the tests except, let's say, cytokine storm, and we attribute the cytokine storm to the presence of certain moiety on the particle surface. If we determine this, we may optimize composition of the particle to still deliver this drug, right, to still achieve desirable biodistribution, but change that property so to overcome undesirable side effect. Endotoxin and sterility, as we reviewed, are important and very challenging to characterize. Sterilization and deparagenation are another challenging characterization steps, and very important to have predictive in vitro models, because it helps to save time and resources and optimize formulation before we enter preclinical animal studies and before material progresses to clinical trials. And with this, I would like to thank my colleagues at Nanotechnology Characterization Lab. Dr. Scott McNeil is lab director. Dr. Steve Stern is the head of pharmacology and toxicology section. I'm very grateful to him for sharing his expertise and understanding structure-activity relationship. Data that I shared with you today were generated by Tim Potter, Barry Noon, Jamie Rodriguez, and Anna Ilinskaya. We have other scientists on the team. I also would like to thank my collaborators from FDA and Eric Simanek from Texas Christian University for sharing triazine dendrimers with us. And thank you all for your attention. I cannot, it's a very good question. So the question was about what is the current stage of the FDA review of the nanotechnology formulations, how many of them are on the market. I cannot give you percentage, like a number of formulations. I can tell you, for example, Doxil is pegylated liposomal doxorubicin is approved for clinical use for cancer therapy. Daunorubicin is Daunazome is also pegylated formulation of anti-cancer drug. Daunorubicin. Abraxane is a nanoalbumin formulation of paclitaxel. There are Ambisome, this is liposomal amphotericin, and there are other formulations with antibacterial products and also imaging agents, such as Feridex, for example. It's the iron oxide nanoparticles. So there is a like quite like a list, several of these formulations that already approved and on the market. I cannot speak about the FDA procedure to approve these materials, but I can say that right now this is a very hot topic in this area. Many of these nanoformulated drugs are getting off the patent, and generic industry starts manufacturing these materials. So FDA has a nanotechnology task force one of the things that this nanotechnology task force is doing, they are researching about the methods to understand bioequivalence and similarity between the brand name nanoformulations and the generic nanoformulations. So I hope I answered your question.
One more question.
Well, the question was how cytokines are inducing multiple organ failure. It depends on the type of the cytokines. For example, tumor necrosis factor, from the name of the protein, you know that it's inducing necrosis, right? So the mechanism is a cell death through induction of the necrosis. They induce hypertension and hypertension. Basically, cytokines are tools that the immune system is using to damage pathogen. And in the absence of the excess of the pathogen, it will do the same damage to the normal tissues in the area where the high level of cytokine is observed.
Thank you.
Thank you.
Hey, how you doing? Okay, and our closer is Pervez Hussain. He got his PhD in India. Subsequently, he did a postdoctoral fellowship in Switzerland. He then came to the University of Maryland School of Medicine in Baltimore. And then NCI was smart enough to hire him. And initially, he was a staff fellow. And now he's a tenure-track investigator. He's head of the Pancreatic Cancer Unit, and his title is Pancreatic Cancer, Current Understanding and Future Challenges. Farewell.
Thank you, Terry. I should use the mouse. Yes. Okay. Okay. I see. Instead of the mouse.
Instead of the laser pointer.
Oh, okay. But I can.
You can still advance, yes, sir.
I can advance from it. Okay, great.
One second.
Okay, good. Thank you, Terry, for the introduction. And what I'm going to do today is to give you and I know it's very hard sitting after 5 o'clock for a 1 hour long session. No, I will finish in 40 minutes, so don't worry. So what I'm going to do today is to briefly give you a short overview of pancreatic cancer and discuss some of the current advancement and future challenges that are needed to achieve the better outcome, disease outcome in this highly lethal malignancy. So let me start with this slide, which shows some of the disturbing facts that are associated with this cancer. It's the 4th leading cause of cancer deaths in the United States. Median survival is less than 6 months, and there is estimated to be 48,960 new cases and 40,560 deaths in 2015 alone. So if you look at the number, if a person is diagnosed with pancreatic cancer, he will most probably die within 1 year of diagnosis. And this is because there is no effective treatment. So, and disturbingly, it is estimated that it is going to be the second, I think this is two. It is estimated to be the second leading cause of death due to cancer by by 2030, which is more disturbing. So what are the risk factors for developing? Yeah, so both. What is happening in last several decades, if you look at the statistics, what happens that the incidence is increasing, but at the same time there is no effective treatment strategy that have been developed. So both incidence and death will increase. And there are several risk factors, and some of the risk factors that are mentioned here: smoking, diabetes, pancreatitis, obesity, and non-O blood group. Smoking is the well-established one among these risk factors, and there are several genetic syndrome and the alteration in the associated genes that enhance the risk of developing pancreatic cancer. And one of the most striking example is hereditary pancreatitis, where you have alteration in trypsinogen gene, and the risk increases by over 50 folds as compared to the normal populations. So what is the status of treatment? And as I mentioned, there has been disappointing progress in the treatment of pancreatic cancer, and I will give you a brief overview that how the treatment strategy has developed. So more than two decades ago, gemcitabine, which is a standard care of drug, was found to be slightly better in enhancing survival in advanced disease over 5-fluorouracil. And since that time, gemcitabine has become the standard care of drug. And then in 2007, erlotinib, which is a EGFR inhibitor, was given in combination with gemcitabine that enhanced the survival only by few weeks, about 2 weeks. But the situation was so desperate that FDA approved that combination drug to be used in clinic. And then recently in 2011, FOLFIRINOX, a combination of 4 different chemotherapeutic drug, showed much better survival, of course, relatively speaking, of about 11 months as compared to gemcitabine alone, but it was associated with a lot of toxicity, and not all the patients could tolerate this regimen. Interestingly, in 2013, as the earlier speaker pointed out about Abraxane or NAB albumin-bound paclitaxel, when it was given with gemcitabine, again, it enhanced the survival by a couple of months, but it had much less toxicity than FOLFIRINOX, and this became like first-line treatment now for the pancreatic cancer patients. And I just changed or inserted some of the new slides because I just found in a recent issue of Lancet, and then this is included here. This is not in your handout, so if you are interested, I can email you the PowerPoint presentation. And here, liposomal-bound irinotecan in combination with fluorouracil and leucovorin extended survival in patients that was previously treated with gemcitabine. So with this cohort of patients that was not benefiting from gemcitabine, this combination enhanced the survival, median survival, to 6.1 months as compared to 4.2 months. And this is approved recently to be used. So as we know, the dismal outcome is because of the poor therapeutic response and also late diagnosis of pancreatic cancer. And there is no breakthrough as yet. And I will give you 1 or 2 examples of the early detection, but there is no reliable marker so far that can be used clinically to detect pancreatic cancer earlier. So what happens that about 18 to 20% of these pancreatic cancer patients are detected earlier and they qualify for resection. So they go for surgery, the resection is performed, and but still in the resected cases, the median survival is less than 2 years. And about 80% of the resected cases, they come back with a recurrence in about 2 years. But about 10 to 12% of these resected cases, they do survive for 5 years, and some of them also survive for 10 years. And there are many clinical prognostic factors that are associated with survival in resected cases. So for example, what is the stage at the time of resection, grading, lymph node spread, and resection margin, if the resection margin is negative or you have the positive resection margin. But none of these are consistent, and we do see variable outcome. For example, there are many patients, stage 1, resection margin 0, perfect surgical removal, but some of them die within a month, within 2 months, and some of them go for 5 years. So that means there is some distinction, like molecular makeup. They are different tumors, although they are a stage 1 or histologically, but it seems that there are different molecular makeup and they are responding differently to the surgical resection and also to the adjuvant therapy. So the question is that, are there molecular differences in tumor determine patient outcome? And I will come back to that molecular subgrouping later on in my talk. So how to improve the outcome in pancreatic cancer patients? It's highly lethal, and there is no diagnostic marker, and there is not an effective therapy that can improve the survival even to 1 year or longer. So we need, of course, effective therapy, and there are several strategies that are being pursued. For example, identification of novel therapeutic target that can be targeted either singly or in combination with the chemotherapeutic drug, and then molecular subtype, as I mentioned, so that a particular subtype can be targeted with a specific therapy, leading to the precision treatment. And then treatment selection based on the individual tumor type. And then one of the latest strategy that is being pursued, and I will give you a couple of examples, how to improve the drug delivery. And I will show you later that when you look at the pancreatic cancer, it has a huge stroma and highly reactive, which is called desmoplastic reaction. And it's hypovascular. There are not a lot of blood vessels. So what has been found that drug is not reaching the tumors. So how to improve that drug delivery? And I will give you a couple of examples. There are many different views which are contradictory to each other. But what has been found that if we re-engineer a stroma and make the drug reach the tumor, that can enhance the survival in the preclinical studies. And also there are 1 or 2 clinical trials that are successful in phase 2 and are in phase 3 clinical trials. So the second thing is, of course, as I mentioned, we have to detect it early, and we have to find the biomarkers that are associated with precancerous lesion, because once it is developed, even in a stage 1, you do resection, people die. So we have to have some biomarker that can detect early on. So for all these things, we need to understand the tumor biology, and I think that understanding tumor biology is key to improving disease outcome. So let me tell you a little bit about how the pancreatic cancer develop and progress. So it has a well-defined course, and it is developed through well-defined precursor lesions. The most common precursor lesions are called pancreatic intraepithelial neoplasia that can be subdivided into 1, 2, and 3 stages, which is carcinoma in situ, and then it develops into cancer. And there are several genetic alterations that are commonly found both in the precancerous lesions and also in the tumor. For example, KRAS mutation is present in almost 95% of the pancreatic cancer. And the most common form of pancreatic cancer is pancreatic ductal adenocarcinoma. So when I'm saying pancreatic cancer, I'm talking mostly about pancreatic ductal adenocarcinoma. And the same percentage of alterations or silencing of P16 is found in pancreatic cancer. You can see KRAS mutation early on in the PANIN1 lesions, and also P16 alterations is found early on. In a later stage of development, you see P53 mutation, and about 50 to 75% of the tumors, they show P53 mutations. And SMAD4 is altered in about 50% of the tumors, and SMAD4 mutation is associated with the distant metastasis and local aggressiveness. So if the tumor shows SMAD4 mutations, you also see a lot of distant metastasis. So as I mentioned earlier a little bit about the tumor stroma, here is the stroma, which we call, which I earlier mentioned, that is a huge stroma. These are the tumor glands, and these stroma consist of stromal cells and lot of extracellular matrix. And these stromal cells, as well as tumor cells, secretes a number of chemokines, cytokines, growth factors, and that help them in a strong tumor stromal interactions here. It's highly heterogeneous, and I mentioned briefly about the heterogeneity and different molecular makeup. So one study early on in 2008 by Bert Vogelstein and Ken Kinsler group at Johns Hopkins, they did the comprehensive analysis of about 27 human pancreatic ductal adenocarcinoma, and what they found that each tumor had at least 68 genetic alterations that can be grouped into 12 core signaling pathways that are associated with carcinogenesis. And in each tumor, they found that a different gene mutation altered some of these pathways. So two tumors, even when they had alterations in different pathways, but the gene that were mutated were different, which showed that it is highly heterogeneous. And in a study by Collision et al. In Nature Medicine, they did the gene expression profiling of a cohort of pancreatic ductal adenocarcinoma. And based on the gene expression analysis, they found 3 different subgroups. They termed classical, quasi-mesenchymal, and exocrine-like. And they found a difference in the survival of these patients with these different molecular subtypes. Similarly, recently in Nature, Wardell and colleagues have published based on the variation in chromosomal structure, and then they subgrouped pancreatic ductal adenocarcinoma in stable, scattered, unstable, and locally rearranged. And furthermore, in another study published recently, a couple of months ago by Moffitt and colleagues, and they based their analysis on the stromal genes. And they categorized as the normal stromal genes and activated stromal genes, and they subgrouped in these types, and they found that the activated type and the normal, they had different survival. If the stromal genes were activated, they have poorer survival as compared to those groups that had normal stromal gene expression. So I will just change gear just a little bit for a couple of slides, because I'm going to tell you that how pancreatic tumors can be subdivided based on the metabolic alterations also. So this is a very complex slide. You don't have to understand everything, but as you know that metabolic alteration is one of the hallmarks in tumors, many different tumors, and so is in the pancreatic cancer. And they heavily used glucose, and they use glycolysis, and some of these important enzymes like hexose kinase and phosphofructokinase, they are regulated by mutant KRAS signaling pathway. Another pathway that is unique in here, that pancreatic tumors use the pentose phosphate shunt only the non-oxidative part to produce ribose phosphate 5-phosphate sugar for the nucleotide synthesis, and it also utilizes a novel glutamine metabolism here that's shown in orange that use GOT2 and GOT1 to produce malate and pyruvate and produce a lot of NADPH. They also use LDH to convert pyruvate to lactate, and lactate is further used in tumors. They also heavily use autophagy to produce building blocks. But what I would try to highlight here that many of these important enzymes, which are marked with asterisks, they are controlled and regulated through the KRAS mutations. And there are many of these inhibitors of these enzymes that are in clinical trials. So I showed you this slide to show this data that is recently published in PNAS, describing again the molecular subgrouping based on the metabolic alterations that they found in tumors. So they found slow proliferating and then glycolytic, lipogenic based on those metabolites. So now let's talk a little bit about that what makes it so aggressive and so therapeutically resistant. So the tumor cells and stromal cells and hematopoietic cells, inflammatory cells, they all contribute to the therapeutic resistance and provide a very friendly environment for pancreatic cancer progression. So let me talk a little bit about this is tumor epithelial cells. So tumor epithelial cells secretes a number of factors, that is platelet-derived growth factors, fibroblast growth factor, activates sonic hedgehog pathway, and that activates pancreatic stellate cells in the stroma. This pancreatic stellate cells is very important in producing a huge stroma and also maintaining that desmoplastic stroma. And this induces the deposition of hyaluronic acid. So you have huge deposition of hyaluronic acid in the tumors that absorbs a lot of water and produces a very high interstitial fluid pressure that suppress already scarce vessel, and this get constricted and the drug doesn't come out of the vessel to the tumors. And I will show you one or two studies that how when you reduce this interstitial fluid pressure by reducing hyaluronic acid, the drug delivery goes in and it enhances the survival in preclinical models. And more importantly, what has been shown that from the very beginning, the immune suppression, the tumor suppressive mechanism due to the anti-tumor immunity is heavily suppressed. So there is an immune suppression because of the heavy deposition of Treg cells, tumor-associated macrophages, and also myeloid-derived suppressor cells. And they inhibit CD8 positive T cells to function. So these several components in the tumors, including the stroma, tumor stellate cells, and the immune cells, they all contribute to the pancreatic cancer progression and therapeutic resistance. So as I mentioned, that there are several treatment strategies, including enhancing the drug delivery and effectiveness of systemic therapy. So target stroma. So one of the preclinical model that has been heavily used in this strategy is this genetically engineered mouse model, where you have mutated KRAS gene and you have active mutated P53, and they are logs. So when you cross these mice with the Cre recombinase, the mutant KRAS is active, mutant P53 is active, and they developed in more than 99% of the mice pancreatic ductal adenocarcinoma, the median survival in 4 to 5 months. The beauty of this model, which was originally developed by Sunil Hingorani, that it faithfully recapitulates the development and progression of human pancreatic cancer. So what they did, they used this model and in one of the earlier studies that was published in Science, and they used the hedgehog inhibitor to reduce the stroma and enhance the vascularization, and they found increase in survival. Here also decrease in liver metastasis in this mouse model. But unfortunately, the clinical trial with sonic hedgehog inhibitor failed. And in fact, it gave the unexpected results, so the clinical trial had to be halted. And this study by Sunil Hingorani that was published in Cancer Cells, using a preclinical model that showed heavy deposition of hyaluronic acid and then deposition of water, and combination of hyaluronic acid and its reaction with water provided the solid mass and enhanced the interstitial fluid pressure and constricted the blood vessel. But when they used hyaluronidase to reduce the hyaluronic acid and reduce the interstitial fluid pressure, it enhanced the drug delivery, and it also enhanced survival in these mice. So this PEG-PH20 is in clinical trial right now. Phase 2b was successful. It's in phase 3 trial. So we will see what happens with this trial. But there are other studies that shown that stromal targeting may not always have beneficial therapeutic response. So what they found is that they used the GEM model, genetically engineered mouse model, and they deleted sonic hedgehog pathway. When they deleted sonic hedgehog pathway, they found decreased survival in these mice. It became more aggressive. Here, when you inhibit, survival is down, and then micrometastasis is up, and also several different precancerous lesions and acinar to ductal metaplasia, they all were increased as compared to the PKC mice. Similar to this study, Raghu Kalori's lab at Houston MD Anderson, they used a different model where they depleted myofibroblast from the stroma, and what they found is that depletion, in fact, enhanced the disease aggressiveness in these mice, and it also reduced the survival. So tumor stromal interaction is complex, and therapeutic approaches targeting a stroma needs caution and may require new molecular taxonomy in the pancreatic cancer. So a stroma has to be re-engineered, but we will have to figure out that what molecular makeup of tumors will have beneficial effect with this treatment strategy. So let me talk a little bit about another very promising target, that is mesothelin. And mesothelin was discovered in this campus in Ira Paston's lab by Ira Paston and colleagues, and Rafit Hussain and Ira Paston have done a lot of studies in on mesothelin and how it can be targeted for treatment. And these are some of the examples which shows that mesothelin is overexpressed in a number of tumors, and they are using the immunotoxin that is targeted towards mesothelin in the mesotheliomas, and also there is a clinical trial that has been initiated to use this immunotoxin that is targeted towards mesothelin in pancreatic cancer by Christina Levine and Rafid Hussain. So I showed that mesothelin background because, again, recently in cancer cell, I think a couple of weeks or a month ago, what Stroms and colleague in Sunil Hingorani did, that they engineered T cells to express the mesothelin receptors. And when they introduced this mesothelin receptor expressing T cells, they lysed the tumor cells in the animal model, and they also enhanced survival significantly. So mesothelin is a promising target that can be used either in immune therapy to target pancreatic cancer and improve disease outcome. So let me mention a little bit about early detection. So as we know, there is no early detection marker that is specific and sensitive enough for early detection of pancreatic cancer. A couple of weeks ago, a paper in Nature by Raghu Kalori's lab at MD Anderson, what they found is that in circulating exosomes, tumor exosomes, the expression of proteoglycan, that is glypican 1, they could differentiate between healthy donors, benign pancreatic disease, for example, pancreatitis, and precancerous pancreatic lesions and pancreatic ductal adenocarcinoma, which showed a high level of circulating exosome that were positive with the glypican. And as you can see, they were highly sensitive and highly specific for pancreatic cancer. So they also tested if this can be used to predict prognosis in resected cases, and they did find that yes, the presence of the decrease of glypican 1 positive circulating exosome can in fact predict better survival in the pancreatic cancer patients after resection. So these are some of the very promising preclinical data and some of the association studies that have the promise, if validated further in different studies, to come to the clinical use. So this is the last part of my talk, and I will take about 10 more minutes to tell you about some of the inflammatory mediators, because many of the inflammatory mediators are found to affect the development and progression of pancreatic cancer in many of the animal models, and also its association have been established with the human pancreatic cancer. So some of the examples are chronic pancreatitis. They are at increased risk of developing pancreatic cancer. You see a constitutively active NF-kappa B signaling. It also enhances some of the enzymes that plays important role in tumorigenesis. For example, COX-2 and NOS2. Terry's interest in there. And many of the inflammatory cytokines, and you do see a desmoplastic stroma, which is highly inflammatory. And you see a stepwise accumulation of the inflammatory changes during the development of pancreatic cancer. The normal PANINs and then pancreatic ductal adenocarcinoma, you see a stepwise accumulation of the inflammatory changes in this model. Also, Craig Loxton showed that although mutant KRAS is important for the development of pancreatic cancer, but it has to be maintained. Just the mutant KRAS is not sufficient to induce pancreatic cancer. It has to be maintained at a oncogenic functional level, that at the pathological level, and that level is maintained through several inflammatory mediators. For example, PGE2. That's what he described in his elegant study, that that high sustained RAS activity initiates NF-kappa B, several inflammatory mediators. One of them is PGE2, and that maintains the enhanced KRAS activity. So one of the interest in my lab, and I will show you 3 or 4 slides, some of the data that that we have generated in our lab, and one of the interest is macrophage migration inhibitory factor. It's a pro-inflammatory cytokine. It is expressed in epithelial and inflammatory cells and many other cells. Primarily, it has been described as a regulator of immune and inflammatory responses. It is expressed in a number of different tumors, including pancreatic cancer. It activates a number of different oncogenic signaling pathway, for example, AKT, NF-kappa B, ARK1 or 2. It also enhances some of the enzymes that catalyzes the reactions that produces inflammatory mediators, for example, COX-2 and NOS2, and it inhibits the some of the p53 function and interferes with the RBE2F pathways. So based on these and several other functions, MIF has been described as a molecular link between inflammation and cancer. So we tested a simple hypothesis that MIF contributes to pancreatic cancer progression and predicts disease outcome. So first of all, we screened the pancreatic cancer patient samples, and we found an increased expression of MIF in tumors as compared to normal adjacent duct in the non-tumor tissue. We also found that a higher expression of MIF in tumors was associated with poorer prognosis. This is the Kaplan-Meier analysis. And this is also the univariable analysis and multivariable analysis, showing that MIF is a predictor, independent predictor of prognosis. And then these studies has to be validated. So we validated in several different cohorts. 2 cohorts are shown here that increased MIF expression in tumors predicted poorer survival in pancreatic cancer patients following resection. So these studies or these findings simply show an association. You have high MIF in tumors, you predict poor survival. So what does MIF do actually? So we did a in vivo orthotopic model, and what we found is that MIF overexpressing pancreatic cancer cells developed accelerated tumor growth and also increased metastasis here as compared to control. And interestingly, if you look at these 2 tumors histologically, 100% of the MIF-expressing tumors, they were all poorly differentiated, very solid growth pattern, as compared to the control tumors, which was well to moderately differentiated. And when you do a gene expression analysis on these tumors, you see that MIF induced a change, a drastic change in global gene expression profile. And some of these genes were associated with EMT, as you can see here, in decreased E-cadherin, increased vimentin, and increased ZEB1. So what does it mean? So recently, there was a paper, and I was very happy about it, that in Nature Cell Biology, David Leiden and Ben Stanger, they published that exosomal MIF, if the exosomes have increased expression of MIF, that goes and prepare the metastatic niche in liver. And if the stage 1 patients who are resected, so coming back to my original point that why some of these stage 1 patients, they die faster than others. So here is some answer that they have enhanced level of exosomal MIF that is preparing their metastatic niche in liver. And they also found that stage 1 patients who have increased exosomal MIF, they show metastasis later on as compared to those stage 1 patients after resection who do not have enhanced level of exosomal MIF. So that's good. That is consistent with what we have earlier described. So what we did, we took the human pancreatic cancers, divided into 2 groups, one with increased MIF, one with the lower MIF. And you see this is the H&E histology here. Tumors with lower MIF, they have well-differentiated tumors, high MIF, poorly differentiated tumors. So the questions we are asking, are there molecular distinction between these 2 tumors? And what is the mechanistic and functional role of MIF in tumor progression? So very talented postdoc in my lab, Shuhu Yang, used an integrative transcriptomic strategy by doing a transcriptomic profile to answer some of these questions. And he tested the hypothesis that MIF regulates microRNAs associated with tumor progression and disease aggressiveness in patients with pancreatic cancer. And what he found that there are several differentially expressed microRNAs when you compare MIF high to MIF low tumors. And some of these microRNAs that are differentially expressed between these 2 groups of tumors, they are also associated with survival. Some of these are mentioned here. So these data were validated in multiple cohorts, patient cohorts of pancreatic ductal adenocarcinoma. And I am just cutting short the experimental part. And what we found that there is a novel MIF-induced signaling pathway which increased MIR301B through PI3 kinase pathway, and MIR301B targets NR3C2. So NR3C2 is mineralocorticoid receptor gene, and its function is not described in pancreatic cancer. And what we found that NR3C2 inhibits EMT. So this axis, MIF MIR301B NR3C2 axis, that leads to the inhibition of NR3C2, enhanced EMT, and enhanced pancreatic cancer progression. And we validated this in the clinical samples. And this is study is under review for publication. So what we are going to do with this data? So we are testing the hypothesis that MIF301B NR3C2 signaling is a potential therapeutic target in pancreatic cancer. So we used the same genetically engineered mouse model, which I earlier described. The tumor expresses a high level of MIF. So we genetically deleted MIF and generated MIF-deleted KPC mice here. As you can see, there is no MIF here. And what we found is that MIF-deficient KPC mice survive significantly longer as compared to the KPC mice with wild-type MIF. It also showed a lower metastasis, as shown here, and specifically in liver. And we isolated the tumor cells from these mice, and you do find that MIF-deficient mice, they have increased cadherin, decreased ZEB1, decrease in cadherin vimentin. This is the Western blot analysis, just consistent with the QRT-PCR data. And what we also found that it has decreased MIR301B. As I mentioned, that MIF enhanced MIR301B. So when you delete MIF, it comes down, MIR301B comes down, and then NR3C2 comes up. And here is some of the IHC analysis for the localization of NR3C2, just to show you where it is localized here. These are the tumors. This is the MIF-deficient KPC mice. So now we have the genetic evidence that when you inhibit in the preclinical model, when you inhibit MIF, it enhances survival, it reduces metastasis. So now there are several ways you can inhibit MIF. There is anti-MIF antibody. There are a small molecule MIF antagonist, anti-MIF receptor antibodies, and also MIF receptor antagonist. So anti-MIF antibody is in phase 1 clinical trial for different solid tumors, and I just found out that it's in phase 2 trial for colorectal cancer and ovarian cancer, and we are talking to the company which has humanized MIF antibody, and if they would like to collaborate with us and give us the drug to start our clinical trial in pancreatic cancer. And there is also in collaboration with Richard Bucala, who has actually developed the mouse antibody and also a small molecule inhibitor. We will be using these inhibitor in the preclinical model and ultimately in the clinical trial. So summary is that a higher MIF expression is associated with poorer outcome in pancreatic ductal adenocarcinoma patients. MIF enhances growth and metastasis of tumor xenografts. MIF-driven signaling inhibits NR3C2 by upregulating MIR301B. NR3C2 is a negative regulator of EMT. MIF deficiency increased survival and reduced metastasis. MIF, MIR301B, NR3C2 signaling is a potential therapeutic target. And I would like to end with this slide, which I showed you right in the beginning, that to increase the outcome or to improve the outcome in the pancreatic cancer patients, we need to work not only on the early detection, but also on the effective therapy. And I think understanding the pancreatic tumor biology and utilizing the biology in developing treatment strategy is a key, and the molecular subgrouping that can lead to the precision treatment strategy for pancreatic cancer will be extremely helpful. So I would stop here and answer your question if you have any, and exactly 45 minutes. So thank you very much for staying. Thanks. Say that again. I see. Yes, yes, yes. So in the resected patients, you can do easily, right? You have the biopsy, you can do it. In the advanced stage patients, that's why it's important, for example, looking at the circulating tumor cells. And there is a recent paper that you can take out from the portal vein without doing so much damage to the patients, these circulating tumor cells. And they have, and I did not mention about that study, it is just published, and you can in fact identify some of the genetic alteration and some of the mutations. So these are the initial studies which I described to you. So now they can do the endoscopic ultrasound and take out the biopsies in those patients, and if possible, and with further advancement, it may be possible to find out in serum or blood or in the circulating tumor cells whether those tumors can be distinguished. Yeah. Sure. That is all. These are very important and very good questions. So now we have to deal with the intra-tumor heterogeneity. So those are the questions that we don't have answer. That how much of the intra-tumor heterogeneity is important to achieve the treatment outcome. We don't know yet. But yeah, all those are important questions. Yes, please.
I wonder if there is any hypothesis about why these tumors are so poorly vascularized. Are there tumors and other cancers that have similar characteristics?
Yes. So we don't know that why they are hypovascular, as opposed to the other tumors. They have increased angiogenesis, and they are heavily vascularized. But one of the hypotheses that a stromal, the huge stroma, and they suppress the formation or expansion of the new blood vessels. But there is no clear-cut answer that why they are hypovascular. Yeah. So, enhancing vascularization and then providing drug is one of the strategy that is being pursued. But we don't know why it is hypovascular, other than the fact that it has very compact and huge hard stroma. When you touch the tumor, it's like a rock. Like I was fortunate enough, I'm not a clinician, but with some of the collaborators, I attended some of the scrubbed in some of the surgery, and I could see firsthand that, and they showed that Pervez, look at this tumor. You will not see this kind of tumor. It's like a stone. And so I don't know the answer of the hypovascularization.
That'll do it, Perez.
Thank you very much.
Thank you.
Thanks a lot.